This is Ecology, and I am your professor, Dr. McAllister. Nice job on completing Exam 1, and welcome to February and the next phase of our course. We spent the first month of the semester learning the foundation and basic concepts in ecology. Now we will explore more complex topics. But I did want to let you all know that I think moving forward, the workload is more manageable. Um, we've got a few breaks built in with spring break, etc. So the material is still difficult and challenging, but I think that you will find that the pace of assignments has slowed down a little bit. So I wanted you to get the really, really difficult and fast-paced stuff done at the beginning of the semester, and hopefully you'll be able to slow down a little bit as you move through this module. So over the next 3 weeks, we're going to discuss population, community, and ecosystem ecology, and then we'll have exam 2. So it's going to happen fairly fast, but like I said, you've got spring break that is also built in there as well. In the prior weeks, we focused more on the organism. Now we're going to go in and we're going to scale up to populations and explore population ecology. A population consists of all individuals of a species living within a specific area. So population ecologists study population fluctuations that are caused by different abiotic and biotic factors. And in order to study populations, we can use different sampling or survey methods, as well as mathematical models and statistical methods to understand population dynamics, demography, and how populations respond to changes in their environments. Studying populations usually begins by determining how many individuals of a particular species exist and how closely associated they are with each other in space or time. So a population can be characterized by its population size, so N, the number of individuals, and its population density, which is the number of individuals living within a specific area or volume. The most accurate way to determine population size is to simply count all of the individuals within an area or habitat, and this is what we call a census. However, this method is often not logistically or even economically feasible, especially when studying large habitats like the entire United States. In some ways, the U.S. Census Bureau tries very, very hard to count all the humans in the U.S., and this is challenging to do, and so they use mathematical models to extrapolate the true population size based on the census numbers they receive. Thus, we usually study populations by sampling a portion of each habitat and using this data to make inferences about the habitat as a whole. There are a few methods used to sample populations to determine their size and density. For immobile organisms, that is, organisms that don't move at all, such as plants, or very small and or very slow-moving organisms, a line transect or quadrant might be used. And sometimes it's a meter square, which you can see through here, a meter square using these PVC pipes that are laid out. Or sometimes it's thousands of meters squared, for example, for looking at forest dynamics, which is used by groups all around the world. For mobile organisms like small mammals, for instance, birds or fish. Mark and recapture is often used. And you can see there is a little trap here that's being used for the recapturing. And this method involves marking a sample of the captured animals with a tag or a band or paint so that you can identify that they have been caught, and then releasing them back into the environment. And later, a new sample is collected, including individuals that were marked. And so these individuals are known as recaptures. Okay, and so when you go back out there and you'll do this, you'll have some recaptures that were marked, and then you'll have some individuals that were not marked at all. We can estimate abundances using simple mathematical expressions, such as, in this case, the Lincoln-Peterson equation. Don't be intimidated by the math. We're going to break it down. And what this equation says is that if you know at least 3 variables, you can always calculate the fourth based on the values of those first 3. And if we look at this equation, it's looking at the ratio between the number of individuals captured and marked in the first sampling event divided by the total population size, so N, okay, and that's equal to the ratio, okay, of the number of individuals that were marked and recaptured in the second sampling event, okay, divided by the total number of individuals captured in the second sampling event. So those are going to be the ones that were marked and the ones that were unmarked. Because of basic algebra, we know we can rearrange this equation to isolate the total population size, or N, because that's what we're looking to know. So therefore, your population size, N, is equal to the number of initially captured individuals, M, okay, times the number of total individuals captured in the second sample, so C, divided by the recaptured individuals in the second population size, so the ones that were marked here. So if you know M, C, and R, you can always calculate an estimated population size, or N. These simple mathematical models also come with some overly simple assumptions. So, for example, mark and recapture assumes individuals are randomly distributed between captures. Okay. Second, there's the assumption that there's no change in the population size between sampling events. So, for example, there's no births or deaths, and there's no immigration or emigration. So, in this case, the population size is not changing, either due to organisms coming in or to those leaving. And then lastly, that marking the individuals with the tags or with paint, etc., does not impact the likelihood of that organism being captured again. So let's unpack one of these first assumptions a bit and talk about distributions of individuals. So species distribution patterns across space show whether individuals of the same species live close together or whether they live farther apart, and this reveals life history characteristics of each species. So, for example, sometimes species distributions are random or uniform in space, but not always. Uniform distributions are very typical of plants that secrete chemicals to reduce competitors. And so you'll see this free area around each plant. And you'll see that they have a very uniform distribution here. Other species have random dispersal strategies, like dandelions exhibit random distributions. And then other times you can see clumped distributions that reflect habitats or dispersal types like gravity dispersal, where the seed falls directly from the mother tree in the plant. And so that's why you'll see these clumped type distributions. Life tables are also used to understand the life histories of different species. And life tables divide the population into age groups, and often also by sexes and how long a member of that group is likely to live or to survive. And these were initially developed by insurance agencies to set prices for life insurance and health insurance, but they're also used or have been adapted by ecologists to describe the life history of any particular organism. Another tool used by population ecologists is something known as a survivorship curve, which is a graph of the number of individuals surviving at each age interval plotted over time. Usually we get the data from this, it's compiled in one of those life tables that we looked at. And so there are 3 types of survivorship curves. Humans and primates exhibit a type 1 survivorship curve that you see here. And here what you see is that there's a high percentage of offspring survive their early and middle years, and death typically occurs predominantly in the older individuals. These types of species usually have small numbers of offspring at one time, and they give a high amount of parental care to them to ensure their survival. Birds are an example of an intermediate or type 2 survivorship curve because birds die more or less equally at each age interval, and that's why you see this negative sloping linear line here. So these these organisms typically have few offspring, and they provide significant parental care. Finally, the third type is typical of trees and marine invertebrates and most fish, which will exhibit this type 3 survivorship curve, because very few of these organisms survive the young stage. However, those that do make it to an old age are more likely to survive for a long period of time. These organisms usually have many, many offspring, but once they're born, little parental care is provided. Okay? So you can think of acorns, for example. There's lots of acorns that are produced, and then they are suddenly on their own. Very few of these actually survive to make it to adult trees. So these offspring, again, pretty much on their own, and they're vulnerable to predation. So for example, for acorns, squirrels and other organisms will be predating on it. But because of the sheer number, of offspring, this ensures that the survival of enough individuals to be able to perpetuate the species. If you noticed, these survivorship curves are distinguished by trade-offs between the number of offspring and offspring survival. So one life history strategy is to live fast and die young, and it's more than just a pop culture reference. It refers to organisms that have many small offspring, but they do not provide resources or parental care to those offspring, which then die relatively young. Examples of our selected species are marine invertebrates such as jellyfish and plants such as dandelions and other organisms that are well adapted to unstable and unpredictable environments. In contrast, K-selected species tend to have larger but fewer offspring and contribute large amounts of resources and energy to each offspring. So elephants and humans, for example, these would be a great example of K-selected species. And these species are adapted to stable and predictable environments. So the trade-offs distinguish life history strategies. So all organisms have an energy budget and allocate energy to different functions like survival or foraging or reproduction, which is energetically expensive. And so it's these energy trade-offs that ultimately maximize evolutionary fitness. Phil Grime proposed this idea of the CSR triangle, which is used for life history strategies for plants. And it characterizes two main factors that limit plant biomass or productivity, those being stress and disturbance. And he proposed that habitats with low stress, so this area down here, and also low disturbance, are characterized by plants with a competitive life history strategy. In comparison, environments in low stress, so still this area here, but high in frequency of disturbance, for here, are characterized by ruderal species. These are short-lived plants that produce seeds very, very quickly, so are selected species. Then in environments with high stress but low disturbance, these select for stress-tolerant plants. So these have life history traits that maximize fitness under very stressful conditions like low water or low nutrients or low light. And finally, Grime argued that there were no viable strategies for an environment of high stress and high disturbance. You can see it there. And so that's what makes this a triangle, because we have no viable strategies for this component here, which would be both high disturbance and high stress. Although life histories describe the characteristics of a population, such as their age structure, population ecologists will also model population dynamics mathematically. And so there are two population growth models that are important to understand because they reveal dynamics about infectious diseases like COVID-19. They reveal dynamics about human populations, and they underscore why some populations are vulnerable to overhunting or overfishing, and why other species like humans tend to be so invasive. So the first of these is the exponential growth model. And this is depicted for populations with unlimited natural resources growing very rapidly or exponentially. And this accelerating pattern of increasing population size is called exponential growth, and it leads to this J-shaped curve. The important concept of exponential growth is that the population growth, or the number of organisms added in each reproductive generation, is increasing. So in mathematical terms, the derivative of this curve, or the slope, is accelerating at a greater and greater rate. And so this is typical of bacteria, for example, where they have a positive intrinsic rate of increase, or R. So the exponential growth curve we were just talking about is possible only when infinite resources are available, and this is simply not true in most natural systems. So exponential growth cannot continue indefinitely due to limited resources. Exponential growth may occur in environments where there are few individuals and plentiful resources, but at some point when the number of individuals gets large enough, resources will be depleted, which slows the growth rate, and eventually the growth rate will plateau or level off. This population size represents the maximum population size that a particular environment can support, and it is called the carrying capacity, or K. You can see right here. So this reflects density-dependent population regulation because when density is high, competition limits available resources, making it difficult to survive and reproduce, which ultimately slows growth. And you can see that this is reflected here in this S-shaped curve. Again, where when you get to the limiting resource, growth is slowing down, and that means that it's density dependent, and you'll have this S shape that is being reflected. So I want you to apply these concepts to the other sections and think about reproductive strategies and how those reflect the life history strategies we explored here. And this week we have one case study, so the clubbed-winged manakin, and explore the clubbed-winged manakin and why evolution could select for worse flight abilities or poorer flight abilities in birds, and then think about how these concepts apply to human population growth. And this week, our assignment will allow us to examine our personal use of finite planetary resources on Earth and compare individual carbon emissions to corporate emissions and what that means for the global economy. And so you'll be exploring this and sharing it as well in an assignment and then also on the discussion board. So I wish you a wonderful week, and I look forward to seeing your contributions.